This is a brief description of our novel segmentation method for Neurite reconstruction from time-lapse sequences using co-segmentation. Here on the top you see a sequence of image stacks at 3 consecutive time steps, where neurons are highlighted in red color. Our goal is to classify every voxel in the data volumes into either neurite or background. Our method exploits the common structure of neuron as shown in the bottom through the co-segmentation principle, which helps aggregate structural information across multiple stacks. Our work is based on the general properties of time-lapse sequence that these images share a similar viewpoint and there are no sudden changes in neuronal morphology, while the appearance of neurons can change dramatically over time. Our co-segmentation framework is composed of 3 main parts. First, the data penalty term is the cost of labeling voxels as neurite or background, independently from the spatio-temporally neighboring voxels. Second, the smoothness penalty measures the cost of assigning different labels to 2 neighboring voxels within the same stack. Last, the tree similarity function H encodes the tree structure coherency of the neurite across image stacks. Our main contributions are the introduction of a neurite model and a tree similarity term. The neurite model tackles branch intensity gaps by incorporating local characteristics, such as shape, smoothness and continuity, while the tree similarity term disambiguates neurite from background, especially at branching points, by enforcing coherency over time. The data penalty term is composed of image and model probability, which indicates the likelihood of regions being neurite. It consists of 2 factors, proximity and orientation. The proximity term ensures spatial continuity of neurites using the geodesic distance over intensity. The orientation term detects smooth paths between neurite parts in an intermediate segmentation result. We calculate this term using the steerable filter at multiple scales along with a vector field convolution. The tree similarity term H enforces tree structure coherency when there is a conflict between paths across volumes, and specifically when a neurite path exists in one volume but not in the other. It is defined as the maximum of the neurite in model probability between correspondences. To generate the correspondences, we match neurite branches by first approximate the tree structures in input volumes using medial axis method. Second, we calculate sample correspondences by thresholding the normalized cross-correlation values between branching points. Last, we align 2 tree structures using iterative closest point technique followed by the Gaussian process regression. These are our segmentation results. The first column shows the collapsed image stacks at 2 consecutive time steps T1 and T2. The numbered points indicate correspondences between the 2 tree structures. The second and third columns show our segmentation results in 2D and 3D respectively. Our method is executed in iterative manner. The initialization is computed by thresholding vesselness produced by the optimally oriented flux method. It requires only a few iterations to recover connected segmentations without gaps, and it is able to handle data with spatially inhomogeneous signal-to-noise ratios.